All right, so I asked you the question, which cell structure in current cells represents that early cell membrane? Let's see what you guys came up, those early micelles. Okay, I see a lysosome. I like that. Mitochondria, phospholipids. Well, that's kind of, not really, but kind of, yeah. Ribosomes, DNA, phospholipid bilayer, fat bubbles. So which, dang, I can't spell to save my life. Okay. The fatty acid bilayer, kind of. Lipid protein membrane. Vesicles I like a lot. Okay. So like the simplest cell structure inside of our cells that would represent that very early first cell membrane would be a vesicle. And then I liked lysosome because lysosomes are a vesicle with a protein in them, right? So that's good too. That would represent the early cell, not the cell membrane, right? And so the vesicle was like the best answer to that question, although a lot of them were still on kind of the right track a little bit. The mitochondria does have a bilayer, and it does have two bilayers, which is a little too complicated for the early cell, right? Now, I asked you to think kind of deep on that one, right? Like deep inside the cell. What the cell, Mr. Mason? It's too early for that, right? That's what you're all thinking. All right, so we're going to move beyond. We didn't get that far? Well, let me go back in time to the origins of life. Right here? Sorry. This is where we are, I think. Okay. So evolution of the protocell, where we left off yesterday talking about one more back. I can. This is. Hold on. We did that. That's where we left off. Okay. So let's talk about this image real quick. That protocell, those very early micelles, these are representative of like those vesicles inside of our cell. And if you throw a protein in them, then they start to represent things like lysosomes. But those very early cells would not have been life forms, right? Those would not have been considered organisms. But they would have been the beginnings of an organism with that phospholipid bilayer or a micelle. So looking at the protocell, the nutrition was the process that would have had to carry on nutrition in order to grow. So you cannot have life without the ability to sustain it and have energy. Now, if organic molecules formed in the atmosphere and were carried into the ocean by rain, simple organic molecules could have served as food. So if you think about all of these nutrients that are trickling down and being formed through Reddy and Operon's experiments, you understand that there would have been lots of organic biomolecules available. So according to this hypothesis, the protocell was a heterotroph, which really makes you think. Did producers or consumers come first? What do you guys think? Who came first, producers or consumers? This is the true chicken and the egg conversation. Who came first, producers or consumers? And I might let this one stew for a minute. We'll see what you guys put. Just see where we stand as a class. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 of you say producers and a few of you say consumers. Not consumers power. Okay. So if the protocell evolved at hydrothermal vents, it could have carried out chemosynthesis, right? Chemosynthesis is when you create energy or ATP using the chemical process, usually in our case involving ATP synthase. But chemosynthesis is a synthesis of an organic molecules by the oxidation of inorganic compounds. And if you think back to NADH and NADPH and the electron transport chain running ATP synthase, do you guys remember that, to create the hydrogen concentration gradient? Okay. If you didn't have me for biology, we might need to review that a little bit. But just understanding that driving that whole process is called chemiosmosis, or chemosynthesis, sorry. So we'll come back to this question in a minute. So looking at nutrition, natural selection would have favored cells that could extract energy from carbohydrates to produce ATP. Remember, oxygen was not available, and the protocell may have carried on a form of fermentation. Now, if you think about that, in this theory, this would actually suggest that consumers would have had to show up before producers, which is really like plays on your mind the way we think about the way we live today, right? Because we depend on producers, but think about this. Things that break other things down and eat inorganic compounds are not making their own energy, right? They're consuming other things. And so if this theory were true, then that would mean that consumers would have came before producers. So looking at a self-replicating system, how do you get more of one thing, basically? So this is the RNA first hypothesis, and we mentioned these yesterday. So this states that the first cell would have an RNA gene that directed protein synthesis, and reverse transcription could have led to DNA genes. So I want to talk about this thing called reverse transcription. There's an enzyme called reverse transcriptase that viruses use to write their DNA into your genetic code. These are called retroviruses. HIV is a retrovirus. It writes its DNA into your genetic code, and then its DNA becomes part of your DNA, and then you copy its DNA onto your RNA, then its codons are going to get read by our ribosomes, right? And we're going to make viral proteins. This is what HIV does. Now, understanding that reverse transcription is a very common thing in the viral world, a virus is not living. It's very simple. It's a strand of genetic material surrounded by a protein coating. It is not a cell. That means it's simpler than a cell, probably a little older than the development of an organism. And so, archaically, it would have had to come first. So we look at things like retroviruses, and we can see them as early evidence of evolution. So RNA was responsible for both DNA and protein formation. And eventually, protein synthesis would have been carried out according to the central dogma. And the central dogma of biology, you guys, is this. We copy DNA onto RNA. RNA wiggles through goes out into to assemble amino acids and make a protein or a polypeptide. You guys, that is the central dogma of biology, okay? That whole process that I just drill into your head over and over again is what this entire study is based around. So the protein first hypothesis, that first one is what if RNA came first? This one now says what if protein came first? The protocell would have developed the plasma membrane and enzymes. Okay, so we understand that, much like a lysosome inside of our own cells. And then DNA and RNA synthesis would have been possible. Now, after DNA genes evolved, protein synthesis would have been carried out according to the central dogma. Again, copying DNA onto RNA and so on out to a ribosome. And after DNA formed, the genetic code had to evolve. You can't see that, can you? What happened? Oh, I don't have I'm powerless today. Is that what you said? I know. I plugged that thing in backwards. I got to blow this up because I can't even read it back there. My eyes are hurting me. What? What happened? Oh, I said I'm not allowed to say blow this up in school. I meant the screen because if you're listening to this on the YouTube recording, you won't see me blowing this up. So, all right, you guys. Just remember, there are many different ways that evolution can be suggested. But we start here at our inorganic molecules being formed at geothermal vents in the ocean, right? So we take these inorganic molecules and we make our monomers, our polymers. Some of them develop into early protocells. Some have the single-tailed phospholipids, again, developing into the double-tailed phospholipids, making what we know now as a plasma membrane in our early protocells. Eventually would have developed into early cells, which would have either contained proteins or RNA, depending on which theory you're looking at, right? Eventually, RNA would have evolved into DNA, leading into what we have today, our actual cells, and a lot of extant or extinct organisms, which are our early ancestors. No, that was the question. I'm going to stop here because I want to give you guys some time because I didn't realize I was going to give you your vocab test. I wanted to finish 8.2, but you guys will be fine to take your vocab test tomorrow. I'm going to stop right here. That way I can give you some extra time.